Usual case for yeah, okay, got it. Always these notifications. Okay, so it's my great pleasure and a special pleasure to introduce today's speaker on the this American Legion Brain Sciences lecture that we started in 1993 with Bernard Mountcastle, the first lecturer. And it's a very special one to introduce Professor Sten Grillner from Stockholm, from the Karolinska Institute. It's a very privileged. First of all, Sten is the capital V world's authority on motor control and brain function. And he has a wonderful book actually coming up in July, Brain in Action, so-called, which is the same title as the lecture today. So Sten received his MD degree in 1968. And from then on, I did also his PhD at, shortly about the same time. And his thesis actually was an eye-opener to the field, because at the time, we thought that everything is controlled by the cerebral cortex, as far as motion, as movement is concerned. I'm amplifying a little bit because, we're supposed formally to start a few minutes later, so I take the privilege to put some background to it. So if I remember correctly, Sten, your thesis, was the focus of the thesis was that if you take an animal or like a cat that cannot walk because it has a spinal cord injury, I think you showed that if you give, dopamine or L-dopa to the cat, then the cat can walk. And that was, to me, was a then very early fellow with Bernard Mountcastle, was really a dramatic demonstration of the capacity of the spinal cord and what the spinal cord can do all by itself, and gradually, Sten pursued that, of course, made the name, the world's authority on spinal cord, function in relation to movement, and with another very, I wouldn't say smart or very intelligent decision to use the lamprey as a model animal for this that has many advantages. Among others, I think you get from the Mississippi River, right, Sten, or something of the sort. It's a local animal. Well, over the years, Sten really, made history with the pattern generators in the spinal cord and developing the whole concept of spinal function as an almost semi-autonomous network that is being modulated and controlled from higher structures like the basal ganglia, which has been his latest, fun hobby, and then the cerebral cortex. So in recognition of all of that, and of course the fundamental contributions that Sten has provided, to the community and science itself, he has been really showered with awards and prizes and memberships in academies. So Sten is, yes, professor of neuroscience at the Karolinska Institute in Sweden. He's member of the National Academy of Sciences here at the United States, the National Academy of Medicine here, the American Academy of Arts and Sciences here, all in the United States. It's very unusual for a foreign member to have all of these elected appointments. He is also a member of the Royal Society of the Royal Academy of Science in Sweden, Norwegian Royal Academy of Sciences. He received probably, always, I think the internal prizes are more sort of important than to someone locally than the external ones. So I see that in 2019, he received the Grand Silver Medal from the Karolinska Institute itself, which is probably the highest honor from his own home institution. And special prize from the King, Royal Prize. Sten is in his capacity, of course, and then tenure at the Karolinska Institute. He was quite involved with the Nobel committee that awards prizes in physiology or medicine. He's been either a regular or adjunct member, advocate member very early in the 80s. In the committee itself, that was a regular member in the 90s and a chairman of the committee of the Nobel Prize Committee for Physiology or Medicine in the 90s. He has received, a lot of other awards that if I still told him before we started, if I'm going to read all of them, it will be half the lecture time. So I will not, I will not proceed with that. But I must say also, as I am, now closing here, that we have been over the years, I think of the rare, close friendships, science and family-wise with Sten and his family. And we have something together, which is a paper, actually, we published in the magazine Science together, co-authored by the two of us in 1989. In fact, it was a nice combination of how reaching, that was my field, was really most probably evolved from modulating spiral networks that have to do with locomotion. I remember the title Sten probably was reaching and locomotion, whatever, so it's a great pleasure finally, because unlike other lectures that I think specific for a given topic and they're all data driven, this one is, I think, this distilled communication of the expert, in the field on brain in action, and we're ready to move, Sten. So we're 12:11, and you have the floor. Thank you very much for accepting to give us this wonderful lecture.
Thank you so much, Apostolos. I really look forward to that, and thanks for your overwhelming description of my efforts, which is not quite the point. But, and I mean, I really enjoyed the paper we wrote in Science because it was a thought actually. We proposed an idea. We didn't do a single experiment together.
Right.
And it's rather well cited. So thanks very much. Now I shall try to get into
Excellent. Yeah, we see it. That's the other advantage of the Zoom meetings. You can see the slides. You sit in an auditorium far away, and many times you cannot see exactly what the slides are. With the Zoom business, it's in front of you. You read them. Great, Sten. Fantastic.
Yes, I'm just trying to, yes, so, okay, very good. I have the images. Okay, so I'm indeed very glad to have the possibility to give this lecture, and thank you very much for the invitation. The title I have given is A Brain in Action, and this little figure here is supposedly now we returned. What happened here? You are screen sharing. I think I have to stop share and then start again.
It's the issue you're having, Dr. Grillner? No. Oh.
No, I think it's, do you see what I have now?
Yep.
So let me start again. So I will talk about the brain in action and evolutionary conserved control strategies. So the brain in action, what it generates is sort of movement and in action. Of course, a whole variety. And the evolution, what I will do in that respect, I will compare the lamprey nervous system, which is the oldest group of now living vertebrates, with that of mammals, to see what is conserved between these two, and the implication is that it did actually exist already when the lamprey diverged from the line leading up to mammals. So what to expect? I will first talk about the building blocks and microcircuits that Apostolos referred to, how the microcircuits are designed and how they are used to generate action. And then I will talk about how they are controlled by the forebrain, and lastly, how the vertebrate motor system evolved over 500 million years and so forth. So when the little baby is born, the newborn is a sweet little survival machine. Is able to perform a few actions. It can cry for help, it can be fed, it's breathing, and a few other things. But during the first year, you have a gradual maturation of the nervous system and the behavioral repertoire until at around a year, you can walk, you can stand, and you can walk, and you can coordinate that. We often say that we learn, that little child learns to walk, but actually it's mostly a maturation process. Long ago, one made experiments on identical twins. One trained one and one did not train the other. And the trained one started after about a year to walk in the morning, and the untrained one started a little bit later the same day. So it's very much a maturation process, which doesn't mean that the child doesn't learn a lot during this time, but these sort of basic patterns of coordination are basically a maturation process. So if you look, I mean, we then have circuits in the spinal cord, as Dr. Georgopoulos mentioned, and we have microcircuits in the brain. The circuits in the spinal cord that coordinates the leg movements are located here, but they are controlled from the brain, turned on and off, etc. And the same applies if you happen to be a fish. It's a spinal cord that generates a motor pattern, and the brain that tells us to do that. For the respiration, you have, of course, respiratory movements throughout life, otherwise you don't survive. And these are coordinated by specific microcircuits in the brainstem. And you have other microcircuits that generates the chewing movements, and you have yet other movements, other motor programs in the spira colliculus, where you can stimulate in specific loci and generate saccadic eye movements in a certain direction and with a certain amplitude. So you have a motor map represented here. And this goes also from lamprey to man. And finally, you have the motor programs that we'll talk about later that display different emotions. What this little guy is thinking is, I think, obvious to everybody. So you have a series of circuits in the brainstem, spinal cord, midbrain, that when activated can execute a particular movement, like swallowing, like respiration, like locomotion, like reaching, etc. And having all these different motor programs, you then also need to have a circuit in the forebrain that determines which motor program should be allowed to be activated at the time. So I will now talk a little bit about these different microcircuits that are responsible for the execution of movement, and later about the forebrain. So the one responsible for locomotion, as we already said, you have a network, a central pattern generator network in the spinal cord that contains the necessary information to generate the walking movements. And you have sensory input that helps to adapt the movements here. So that resides in the spinal cord. But you have the command centers in the midbrain brainstem, the mesencephalic locomotor region, MLR. And when that is turned on by reticulospinal neurons, it will activate this. So this is the circuitry required for the execution. Now, the forebrain is involved in the control. We have both the basal ganglia, which has an output with tonic inhibition. So the inhibition must be lifted if locomotion should be generated. In parallel, we have cortex that provides input to striatum, but also at this level. So essentially, what one can say is that this structure is conserved from lamprey to man. And the circuits, if you then, with the next level question, not only that you have the circuit, but how are they designed? That has been, the analysis has been helped by results from the Jessel laboratory showing that you have discrete modules of genetically specified subtypes of neurons that are involved in different movements. So you have these excitatory neurons that in the mouse generates the rhythmic burst pattern, and you have coordinated movement that helps to coordinate walk, trot, and gallop. And in the lamprey, you have, sorry for the telephone that I suppose you heard also. We looked at this, and it's rather detailed, elaborate analysis if you shall understand how a network like this works. You must first identify the cells. You must identify the cellular properties of the cells, and you must identify how the cells talk to each other. And what we then know that when these circuits are turned on from the brainstem, then they can generate burst pattern. And the coordination between left and right is set by inhibitory neurons. When you have all the numerous details required experimentally, then it's also helpful to use simulation as a tool. In order to see whether all the different pieces of information really make sense and can produce a motor pattern. And what we did many years ago was then to model each type of neuron and the correct number of neurons with the correct synaptic activity, etc. And if you then activate these with the reticulospinal neurons from the brainstem, and you drive the excitatory and inhibitory neurons. With the experimental data available, we could generate the motor pattern. Moreover, we could generate the motor pattern in the normal range that the animal swims, from 0.1 hertz to about 10 hertz. And that gave us confidence that we had a circuit that generated this. So I like to emphasize that simulation together with biological detailed data is a very useful tool. So this is a basic circuit and the basic design, and it applies to the lamprey which is other fish, salamanders, the walking pattern of birds, and the flying pattern of birds in terms of coordination between the wings in phase, like a gallop of the cheetah. And you have variations here, and you have variations even here. But the basic design of the locomotor pattern is coordinated in the same way. So it was a good invention early on that generated that. If we go to another CPG, we have the brainstem CPG for respiration. That's located in the brainstem in the pre-Bötzinger complex, consists of inhibitory blue neurons and excitatory glutamatergic neurons. You can block the inhibition and you can still generate the burst pattern. And the burst pattern is generated by a subtype of excitatory interneurons and then drive another ones that then activate the motor neurons. So this is essentially the core of the respirator. But it's modulated, of course, by chemoreceptors that determine the rate of respiration, lung receptors, etc. And this generates the inspiration. Then we have another center that generates expiration, and that is inhibited by the pre-Bötzinger complex. So they alternate, so inspiration and expiration alternates. So that's the basic design. Of course, this doesn't tick on by itself. It is modulated by a whole variety of different things, as you can see here. And it's even clearer in this. So you have the pre-Bötzinger complex, but it's, of course, regulated by vocalization and speech. As I talk to you, I regulate the airflow and the shape of the oral cavity that produces the sound. And you have sighing, which are related by peptides, that is important for homeostatic mechanism. When you swallow, it's not a good idea to breathe at the same time. You have other small central pattern generator CPG networks like licking, like chewing, like whisking, and so forth. They are independent, but they are coordinated to some degree with the respiratory movements. So it is a whole set of different things. And the respiratory work is pioneered by Jack Feldman, and the things around here, particularly the Kleinfeld laboratory, has been important. When you have chewing, that's a separate CPG, and there you have a network in another part of the brainstem with excitatory and inhibitory interneurons that also is modifiable in different ways, that have different parts that you can chew on one side or the other side, and which then regulates the chewing movements with the different motor neurons. And that, of course, turned on from the brain. So that's essentially chewing. So that's another example. Yet another microcircuit that is used for reaching, when you reach out to an object, and then when you like to grab, you need first to extend the hand and then contract. As you can see, the frog can do this, even particularly when it's attacked. Birds, of course, do reaching very precisely and with great capacity to maintain postural stability, etc. And you have the reaching movements in primates. This is an image actually from a paper by Dr. Georgopoulos, where it shows the reaching movements at different points. And what was pointed out also was that when you walk in a complex terrain, you need actually cortex to with precision place the limbs like for this cat. What we pointed out in the paper that was mentioned before was that actually reaching towards an object, and this is a very similar type of coordination. But this is a little bit more difficult as you reach out here. And it was later shown that it's actually the same neurons are used both for reaching in this condition and for reaching out. So what a few years ago was shown by the Arbor laboratory was that you have a microcircuit in the brainstem, the medullary part of the brainstem, that contains 3 subtypes of neurons. One subtype projects directly downstream, and when you activate that optogenetically, you can produce a reaching movement. You have another subset of neurons that when you activate them, open up the hand, which is of course required if you are going to grasp. And then the third category that produces the grasping combined with a hand-to-mouse movement. So you have this reaching, grasping brainstem microcircuit also in the brainstem that takes part of the coordination. And the cortical commands for reaching, as Dr. Georgopoulos pioneered now many years ago, showed that if you had monkey pointing to in different directions and you recorded neurons, the neurons in the movement direction would be most activated, but also the neurons in nearby positions like this and that would be co-activated, but to a lesser degree. And it was shown that the cosine function that does that. And conversely, the neurons would be inhibited by movements in the opposite direction. So that's a cortical command for reaching that may use the brainstem circuits. Another set of microcircuits command centers is a preaqueductal gray that is located around the aqueduct between the third and the fourth ventricle in the midbrain. And you have different compartments. I should say that it's rather big structure. In humans, it's even 23 millimeters long. But there you have discrete compartments. One, when activated, leads to escape. Another to freezing movement, so you just stop, which is good for protection or sometimes. And you have one that elicits fighting behavior, aggressive behavior. You have another set that activate vocalization, threatening vocalizations, etc. You actually also, as Sten Pfaff has shown, you have sexual behavior that is conveyed from hypothalamus by the preaqueductal gray. And you have maternal behavior initiated from other parts of hypothalamus that is also mediated here. So it's a complex hub that then further utilizes the brainstem. If we take the escape part, the DPAG, it triggers locomotion, fast locomotion, via the locomotor command centers and mediated to the locomotor CPG. But you also have input from a variety of things like superior colliculus, cortex, and of course hypothalamus and basal ganglia. So, and the same similar type of schemes you could put here. So you have these different schemes. And finally, as I mentioned before, expression of emotions. If I can move that along. Darwin, in a very nice and impressive book they call The Expression of Emotion in Man and Animals. He thought about whether this was innate or learned. So he then could show that children born blind was smiling as nicely as seeing children. And crying, of course, occurs directly after birth. You have other expressions like being worried, disapproving, of course, being very angry here. He concluded that people around the world, he was writing to missionaries around the world and inquiring about whether in all parts of the world one was smiling, etc. So he concluded that these were innate motor patterns and not, of course, at this time, genes didn't exist, neither DNA, at least it was not known. So of course, and he did furthermore, actually, based on other people's work, showing that you could elicit the different expression by stimulating different facial muscles, combination of different facial muscles. And so is in a sense, he conceived the idea of a CPG activating the different motor neurons, if you like. Anyhow, then, of course, in different cultures, what you laugh at and what you cry for and what you're worried by is, of course, culturally bound, but the motor expression is innate. So, returning to the figure that you just said, it makes a further point that you have this motor pattern, and you actually also have a very similar organization in the brain. So the execution of movement is mostly a midbrain brainstem spinal cord affair, and is dependent on dedicated circuits for each behavior. The planning and initiation of movement, on the other hand, is a forebrain affair, the cortex, basal ganglia, the dopamine system, and other structures like that. One can make the analogy of an orchestra. The microcircuits is as the single microcircuits for different patterns of behavior. Each know their own thing. They can perform if it's reaching or if it's playing the violin. So this is like an ensemble of microcircuits, if you like, the analogy between the orchestra and the midbrain. And the director, the forebrain, determines when each microcircuit should be called into action and stop. So it's a director that controls the orchestra. It's like the forebrain controlling. So the structure for execution can be simplified in escape, in hypothalamus, sort of. You have the downstream motor centers that they can be called into action. You have the command centers in the periaqueductal gray that is also influenced from hypothalamus. So this is more or less machinery for execution of different actions. And you have cortex and the basal ganglia in control of these. Cortex for the direct excitatory control, and the basal ganglia receives major input from the cortex, but it also tonically active inhibitory. But it's if you lift the inhibition that action can come about. So I shall first talk briefly about the basal ganglia and cortex, and then the evolutionary aspect. So essentially, the basal ganglia in the forebrain is composed of the output level, the GPIs, SNR. It consists of inhibitory GABAergic neurons that are tonically active. And they project to the different motor centers in the brainstem. So they are kept under resting condition, all in control through inhibition. And the motor centers can generate saccadic, locomotion, posture, swallowing, etc. And what is required to initiate behavior is, of course, to inhibit the subset of neurons that control the specific behavior. And that achieved from neurons from striatum. They are difficult to activate, but when they are activated, they project to specific subsets of neurons here. And this activity is facilitated by dopamine. But that's not the whole thing. It's also, you have another subset of striatal projection neurons that instead prevent movements or have a negative effect on movement by, they have dopamine receptors, and they, when activated, they inhibit the tonically active neurons here that in turn disinhibit these and then enhance the inhibition of these two. So this is, you have a no-go pathway. In a simplified version, you have a direct go pathway, and you have other pathways that also can mediate the excitation. So that's the very general scheme. But it's really too general, because a recent study showed that if you take the output stage of the basal ganglia, it consists of a large number of neurons that target no less than 42 different targets in the brainstem. That can be reduced here. So you have some neurons that target the inferior colliculus that can relate to locomotion. You have different targets here in the superior colliculus and medullary reticular formation, and so forth. So you have a very detailed projection pattern. It's like the keyboard of a piano where you can elicit different movements. Or I mean, they are all anatomically inhibiting, and in order to have action, you need to block their activity. Another important thing is that all these cells also send a branch to thalamus, and then with information conveyed to both cortex and striatum. It's sort of efferents complete. It keeps the brain informed about which commands are issued here. Another very recent data that has indicated very strongly the very specific organization. It's not a diffuse organization, it's a very, very specific. You have in striatum here, you have specific areas in the somatomotor part here for the forelimbs, for the hind limbs, for the trunk, for the jaws, for the inner part of the oral cavity. And these are discrete, specific compartments. And if you look on the cortical projection, it's specific cortical areas that projects to specific parts in striatum, that projects to specific parts at the output level for the direct pathway. But for the indirect pathway, we also have have specificity. So the different subtypes of neurons here in pallidum then projects back to the same general area here. So it's an previously unheard degree of specificity. And then the information back to cortex and actually also striatum is kept very precise. So the image that we get is a very well structured modular control from cortex to the basal ganglia to the output. And that Foster and the Dong laboratory actually a result of the US Brain Initiative. So the relation between cortex and the basal ganglia has been described by Arber and Costa as the cortex may express a wish, while the basal ganglia determine whether the wish will be fulfilled. Paraphrasing a little bit. So what about the general image of cortex? Well, first let's say that cortex provides the input to striatum. So about 50 to 60% of the input is from cortex, but it's significant input also from thalamus that conveys information from cerebellum and other structures. And also there's the dopamine, 5-HT and histamine, etc. So if we think about cortex and what we can say is that if you stimulate not with single stimuli but with a little bit longer stimulations, you can actually elicit integrated movements, as the CAST laboratory have shown in Graciano. So essentially, in the parietal lobe, you have different compartments that can elicit, for instance, any of these. And the green here, the defense region, or you can elicit the reach here. And the reach is then media is sort of projecting to the motor areas, both the motor cortex and the premotor cortex. So you have different subcompartments here that generate these different integrated patterns of behavior. This is from primates. And what is important in this context is that there is a major projections also to striatum. So you have a reaching, you have a defense area, you have a grasp area, and you have a face area. So this is again emphasizing the precision and the modularity. If you then look on the projection pattern from cortex in mammals and lamprey, you have projections to the optic tectum, you have to mesencephalon, tegmental, vertical spinals, and to the spinal cord. That's a motor part directly. And then you have the input to striatum, but also the other parts of the basal ganglia. And you have input to thalamus. So you have this projection pattern in cortex and actually have this both in lamprey and mammals. So the output from cortex from layer 5, and it's from, of course, the pyramidal tract neurons that goes through striatum, provide excitation there, but then goes down to the different motor centers. But you have also a group of cells in equal number that called the intratelencephalic. They project, they excite the PT neurons, and they interact at the cortical level. But they say also a very major projection to striatum and with a rather broad pattern of excitation. So that emphasizes again, that is, I think, very difficult to understand cortex without the basal ganglia and vice versa. So now, with that sort of more general thing, I will then ask the question that I mentioned before, to compare the lamprey nervous system, how that is organized with that of mammals, with the conclusion that was that this was about 500 million years ago. So the lamprey it's has a frenetic important position in evolution. It diverged from the main vertebrate line some time ago, and it developed all basic features of the vertebrate nodes except cerebellum. And there are, as you can see, no jaws. When we started these things, this little part here is what becomes cortex. And it was assumed that it was entirely olfactory. So we were very surprised when we started to stimulate that we could elicit discrete eye movements, discrete orienting movements, discrete swimming movements, and also oral movements, activity of the mouth, which is of course also very important. We could, so there is essentially a motor area of pallium or cortex. And we could show that you have projections downstream. So I'm just showing this slide again to emphasize that you have the projections of the optic tectum, it's glutamatergic with similar somatic properties, and to the basal ganglia and thalamus, as we have in our own brain. And we have these two subtypes of neurons, both in lamprey and mammals. And now, what about the structure? We could show that cortex is three-layered. It has a molecular layer, a cellular outer, and an inner cellular layer. And we could show that about 20% are inhibitory neurons and the other are excitatory neurons. And that is the same proportion as in mammals. We then recorded the PT neurons that projected to the optic tectum, and you can see that they have a cell body and they have dendrite that diverged out to the molecular layer, as in mammals, but the dendritic tree is much broader. So it's picking up information from a broader area. These neurons have dendrites and they have spines on them, as we have. And we have two types of neurons, the sort of regular spiking neurons and burst neuron that you with brief stimulation gives information to. So these, in a general sense, these neurons are similar. What is the sensory representation that you have in the lamprey cortex? What we could show is that we have a retinal representation, a retinotopic representation, where if we stimulate different parts of retina, we can record in cortex a response, and different parts of the retina projects to different parts. So there is actually a structure that can be equated with V1. Is it a somatotopic representation? We can show that actually activation of trigeminal activates one area of cortex and dorsal column spinal cord another area. So what we could show, to our great surprise, was that there is a visual retinotopic area, it's a somatosensory area, and there's a motor area. So it's the same sort of basic design as we have here. So what we conclude or propose is that the dorsal pallium of the lamprey corresponds to the dorsal pallium of ancient reptiles, and which in turn then developed to neocortex. You have the line that goes to dinosaurs and birds, in which you have further changes. One should note, however, that birds, ravens, etc., are as smart as monkeys or even apes in behavioral tests. So the basic levelings of cortex is as conserved among vertebrates. So what about the basic ganglia? This figure you have seen, and we have then compared lamprey and mammals in terms of cell types, input to stratum. And as you can see, it's very similar. We have the same type of neurons, the D1 and D2, stratum projection neurons, spinal dendrites. These neurons are difficult to activate. They have inward rectifiers neurons. They are GABAergic. They contain DARP32, a master switch molecule in both mammals, and they are hyperpolarized at rest. The striatal interneurons, we have cholinergic and fast spiking interneurons, but we don't know if we have the other more recent subtypes of neurons. You have the output pattern, the output pathway with SNR and GPI, spontaneous activity, etc. And you have also the indirect loop. Yes. So it seems it's a very similar design. So to reiterate, you have the D1 substrate, you have the D2 stratum projection neurons, the indirect pathway, you have the inward rectifiers, you have the DARP, you have the synaptic input with similar properties, you have the subthalamic area and glutamatergic area, and they are spontaneous, active, and rebound. So to reiterate, this is the lamprey stratum, and you have the input, the different as in you and me. We have pallium and cortex. You have, as I said, the spiny dendrites, and we have also the expression of the same molecules as we have. This is just an example to give. On the output side, we have the GPI here. They are spontaneously active. And if we block glutamatergic transmission, they are active. So that's the same thing as we discussed for mammals to control the different motor centers. And we could also show that different parts of different neurons at the output stage project to different structures, and that's of course required if they should contribute to the control. So at rest, the pallidal output of the brainstem is tonic and inhibitor, and is removal of the inhibition sufficient to generate the motor pattern. And what you can see here is you have striatum pallidum and then the locomotor command center. And if we block the GABAergic transmission here, as we do with the GABAcine, then we can initiate alternating activity. And if you have the body intact, swimming movements. So the tonic inhibition is important in controlling whether a motor part should be. The dopamine system is also important, and it's the same story here with lamprey and rodents. It's the same coupling scheme. The dopamine neurons inhibit stratum, they inhibit the subthalamic nucleus and SNR, and also downstream motor centers, both in rodents and lamprey. On the input side, you have the input here, salient pathways. You have the peduncular ponti in the lateral habenula, you have other types of sensory information that fits. But it's all there. And what one can show here that salient visual stimuli, electrosensory stimuli activate the dopamine neurons, and which will then modify to facilitate dopamine transmission in the lamprey, just to show that we have D1 receptors. And if we record from the cell and apply dopamine agonist of the D1 type, the cells do what they should do. We have D2 receptors in situ. And if we apply D2 agonist, we instead have inhibition as in mammals, and we have a projection pattern. Then asking the question, if we denervate the dopamine cells with a toxin like MPTP, what symptoms do we get? And we get Parkinson-like symptoms in that then will be very difficult to elicit locomotor activity, and spontaneous locomotor activity is practically reduced. There's some rigidity, there's some delayed reaction time, and so forth. And we can show whether we can recover function if we give dopamine agonists. So our sort of conceptual scheme is that we have these different modules in stratum that control different basic patterns of behavior, like locomotion, eye movement, steering, etc. Whether these that are conserved, and they contain all these sort of different matter patterns. When they are if they are turned on by activity in pallium cortex, but thalamus can contribute also. And the dopamine system is very important in determining the responsiveness. When the dopamine system, the D1s are enhanced and D2s suppressed. So this is a conceptual scheme for the lamprey. What happened during evolution? Probably, since it seems that one has retained the same basic pattern, innervation pattern, what probably happened is that during evolution one has created progressively larger number of modules that can contribute to the more complex behavioral repertoire that develops. So it has been a conserved design that apparently has contributed well. So it has been modified, had not been modified, but instead you increase the number of modules. That's the conceptual scheme that we apply. I will not be able to talk about that, but we have also simulated to try to understand stratum and the operation of stratum. We have simulated it. We are simulating it in great detail with the appropriate properties of single cells, etc., the connectivity scheme, etc., which I could have delivered a lecture on by itself. So the vertebrate phylogeny. So what have we concluded? We have concluded that there's a 3-layered cortex here, 3-layered cortex there, and this is 6-layered cortex. The basal ganglia was there, of course, very much fewer neurons, but essentially the same components. The dopamine system is very similar. So it has the same type of neurons, the same type of projection pattern, the same type of input. And habenula, which I have not talked about, is also resembling that. So, summarizing, we have the system for execution with the microcircuits that are called into action and controlled by cortex and the basal ganglia. But the basic coordination is to a large degree decided by the microcircuits. So what it all means is that at this point of evolution, some 500 million years ago, the blueprint, the plan for our nervous system had already been conceived by evolution. Of course, a lot of additions have happened during this period. You have added limbs, you have added a lot of things, but the basic blueprint of the organization of the vertebrate nervous system was conceived very early and have of course been elaborated, and of course with a large increase of number of neurons that gives more processing capability. So let me just pay credit to my dear colleagues that have been very instrumental for all this. Peter Wallén for the CPG work, Jeanette Elgren Kataleski, Professor in Neuroinformatics, has been important. Johanna Frost-Nylén, Britta Robertson has been very important for the entire morphological analysis. Andreas Kardamakis, Joanne for the dopamine work, Marcus Stevenson-Jones quite some time ago now for the basal ganglia work, Shreya Sriyanarayana for much of the pallium work, and Alex Kostler for a long time contributing to the simulations of lamprey, and then currently Stratum and Johannes Hjort. And there are more to come. Of course, a special credit to the lamprey itself. So this is simulations I have not spoken about. You have a simulated lamprey with a simulated CPG with a basal ganglia swimming towards blue objects and avoiding red objects. Just as a finishing thing. So thank you very much for listening. And
Well, thank you very, very much, Sten. It's a wonderful tour de force. Well, I don't know, I would say let's look at the future, but I don't know what the future will be. I mean, you uncovered everything practically as far as the motor system goes. And I don't know whether you plan to go to vision or other kinds of challenges.
But no, I think, I think there is lots of things like visuomotor coordination is an unbelievably complex thing. And so that's
wonderful. That's wonderful. And it's so nice to see, of course, the combination of technologies, electrophysiology and anatomy and genetics and tracings and simulations. And well, it's the bottom line is that you need all of them to get an understanding of what's going on in a very complex system. So many thanks. As I mentioned, the lecture is being recorded. So in our site, it will be Sten's email, which is very simple these days to remember, is sten.grillner@ki.se. And he agreed to respond to emails, right?
Absolutely, with great pleasure.
Yeah, that's wonderful. That's wonderful. To end, also to point out that the two institutions, the Karolinska Institute and the University of Minnesota, have been long associated, through, I think, some funds that are provided by Carlson. And actually, some meetings, both here and in Stockholm, just between, we're not exactly sister or brother universities, but we are certainly on the same wavelength. So that continues the tradition by accident, if you want, but it's very nice to have another joint venture or opportunity between the two institutions. Thanks again, Sten. Appreciate it a million. Thank you very much.
Thank you so very much. I have very much been very grateful for this possibility. So thank you. And let's maintain the Minneapolis.
I think we should maybe we can have a Zoom meeting of the two institutions, select the themes. We used to have the pain, the motor, there were groups of joint interest. That's something we can have some sort of interaction without necessarily having to travel. Well, this concludes this year's lecture.